The pathophysiology of ischemic stroke. Welcome, and thank you for joining us on this incredible voyage to understand the third leading cause of death in the industrialized world, the #1 cardiovascular cause of death in China and Japan, a leading cause of adult disability in the United States, with a total estimated cost to society of $53.6 billion a year. Stroke. Stroke is caused by a transient or permanent interruption of blood flow that leads to ischemia and triggers a complex series of pathophysiological events that causes injury to the brain. The consequences of these cerebrovascular events for the patient may be death or disability, with the loss of productivity and a decrease in quality of life. The lack of effective acute stroke treatment is a major health burden for patients, their families, and society in general. The pathophysiology of an ischemic stroke involves a complex cascade of events that occurs within the brain. The human brain requires a constant supply of oxygen and glucose, which are used for the production of energy. These substrates are delivered through a vast network of blood vessels throughout the brain. A variety of vascular events, including plaque formation and rupture, can create an embolus or clot. An embolus moving through the vascular system can occlude a cerebral blood vessel, thereby reducing the delivery of nutrients to the affected area. The area receiving little or no blood flow is known as the ischemic core and is comprised of cells that die rapidly. Surrounding this ischemic core is an area of reduced blood flow called the ischemic penumbra. The fate of the ischemic penumbra is dependent upon key events that determine life or death for cells in this region, since cells in this area are in danger but not yet irreversibly damaged. Within the penumbra, the reduction in energy substrates leads to energy depletion and creates an ionic imbalance that causes cell membrane depolarization. Excitatory amino acids, including glutamate, are released in pathological quantities. Energy-dependent reuptake of glutamate is diminished, allowing for greater accumulation of the amino acid in the extracellular space. Continual activation of glutamate NMDA and AMPA receptors contributes to intracellular calcium overload. Within the cell, the increased calcium activates a series of enzymatic reactions, including the activation of phospholipases, which degrade phospholipids, causing the release of arachidonic acid, the production of nitric oxide by the calcium-dependent enzyme neuronal